Good morning. Welcome for the second day to this European Coral Reef Symposium. It looks that the second day is a little bit harder respect the first one. But anyway, before to start with the second plenary lecture, just a technical communication. After the plenary, we will meet in the theater outside for the group picture. So before to start to move in the different room, move outside for the group picture. So this morning, I have the pleasure and the honor to introduce Sergio Rossi, the speaker of this second plenary lecture. Sergio is who knows Sergio knows perfectly that he's a very passionate scientist, and he focused his research on global change indicators, coastal benthic population, marine vertebrate ecology, and marine conservation. His dynamic nature and drives him in this last period to lead two significant projects, European projects. One is the coast action on marine animal forest, and the second is another European project, an Horizon project called Ocean Citizens. He began his career at CSIC in Barcelona, and he spent nearly 10 years as a senior researcher at the Università of Barcelona. Currently, he is in Italy. He is a professor at the University of Salento, but is also a permanent professor at Labomar in the University Federal do Ceará in Brazil, and is also the scientific director of Underwater Gardens International in Barcelona. But Sergio is not only an important and well-known scientist, he's also a prolific science journalist. He contributes to work with various journals and magazines. He has authored also eco-thrillers and the children's book. This is a very important component in our job, in my opinion. And his notable works include books like The Blue Planet, A Vanishing Universe, The Forgotten Continent, Vision about Antarctica, and his book has been translated also in English about Oceans in Decline and A Journey in Antarctica. Very nice book. I have read some of them, not all, but okay. I think now the floor is yours. Thank you, Sergio, for being here and for sharing with us your insights.
Thank you very much. Thank you very much, Carlo and Massimo, to invite me to this, to make this talk. I'm really very happy about that, and very honored. Also to all the committee that invited me to this European Coral Reef Symposium. I will try to be brief. I will really try hard, but the things that I have to say today are just a little bit complex, even if they are presented in a, I would say in a soft way, okay? But all of us have these ideas in mind of what we should do and where we are in this moment. So we all know this graph. In 2017, we have this paper, okay, of Nash, which started around, among other papers, to talk about the limits of several things that are happening, the synergistic effects basically on the Earth, from overfishing, pollution, exploitation, soil exploitation, climate change, and so on and so on. I just put one here because, as you know, there are others that are increasing because we have more data and the things are fast, let us say, change very fast in the world. And I don't want to depress you in the first slide. The second slide is a little bit more depressing, but it's very nice. I love this slide from Daniel Pauly. Daniel Pauly published it in 1998, okay? And in this slide, basically what you see here is the oversimplification of the ecosystem, what is happening now all over the world. For several reasons, okay? The things are fastly changing. But one detail here has to be accounted. The thing is that Pauli, in 1988 paper, he put only the fishes, put only the pelagic organisms, only the sharks, tuna, gropers, and other organisms that were overfished. But then he realized that something was missing. And the thing that was missing is the oversimplification of the ecosystem engineer organisms that you see here, okay? The amount of phosphorus, the amount of nitrogen, the amount of carbon is exactly the same, of course. But the thing is that everything is fastly changing in the world, in the sense that we have all these, processes, all these fluxes changing very, very rapidly now. So the organisms that are living and taking advantage of fast-growing species are, based more on microbial loops, more on fast-growing species like, for example, medusae, or for example, small fishes, or for example, hydrozoans and other organisms. This is what Pauli wanted or intended to project. Because this organism, we are losing this organism in some way. This is an ecosystem engineer. Jones said very well in 1994 in this Oikos paper that I really, that's a really important paper in the ecological literature in which he described these organisms, trees in this case basically, but he also talks about coral reefs, even if it's in a small piece of these papers, as facilitators, organisms that are essential for complex dynamics, for complex relationships among the organisms that were there. And we have this on land, but why not having this also on the field of the sea? So on the bottom of the sea. Take into account that on the bottom of the sea we have marine forest. The same kind of forest, but completely different because, of course, the fluid is different, okay? We have in the benthos, from shallow areas to deeper areas. We have vegetal forests that are basically kelp forests, seaweeds, or maybe seagrasses, like, for example, Posidonia oceanica or Thalassia destudinum. And we have animal forest, in which you have organisms that are basically dominated by metazoans. In 2013, I published a paper in which I just wanted to describe a little bit what is this marine animal forests are about. And I said, the animal forests that live in three-dimensional structure similar to a vegetation forest, but composed basically of sponges, cnidarians, bryozoans, ascidians, and other benthic suspension feeders in the ocean benthos. It's okay. But my purpose here was not to put, this kind of, let us see, effective word or effective 3 words in the, in the plate. Was breaking silos. So what I mean with that? Very simple. With marine animal forest, we can gather every habitat that is composed by basically metazoans. So more than 2,000 pages, more than 60 chapters, and more than 120 specialists in 4 different volumes, 2 books, the first and the second, were gathered, and people that was working with cold water corals started to see in the same book people that was working with sponge grounds, or people that was working with bryozoans. So the people started to realize that this may be a common platform to fight for conservation for these organisms, and also, even better, to understand these organisms. So why they are so important, the forest of the sea in general and the marine animal forest in particular, which is the speciality that I'm bringing here? Well, you know that they are barriers for hurricanes. The forests of the sea are key habitats for the first stages of life. They feed and shelter fragile species. They transform the hydrodynamics. The forests of the sea are also biodiversity hotspots, and they also immobilize carbon and are part of the biogeochemical cycles. And also, again, this is also for me very important, they may be sometimes for the local people and for the indigenous people, very important parts of their lives. When I have been working in Brazil, for example, with coral reefs, with seagrasses, and especially with mangroves that are not marine animal forests, of course, but are forests of the coastal areas, I realize that they see that these forests are more than regulating biogeochemical cycles, or are more than carbon sinks, or are more than giving biodiversity hotspots. For them, sometimes are everything. That was the word that they described when we make the paper in which we try to understand what does it mean for them, the forest of the coastal areas. And they say everything. Everything is everything. So there is also a more human, spiritual, and cultural part that sometimes we are neglecting, but it's also important. Many threats are just impacting the marine animal forest in general. I will just highlight two, okay? One direct and the other a synergy of an indirect, let us see, impacts that we have. The first is bottom trawling. Bottom trawling is the most devastating by far of the animal forest of the world. In 2002, Dayton and Thrush published a paper in which they demonstrated, there were, of course, rough numbers, that 2002, every year we bottom trawl 1/3 of the continental platform. And that's the truth. So there are no opportunities for the organisms that are living from shallow to deeper zones where we are making bottom trawling. And we are making bottom trawling more and more and more deep, deeper and deeper and deeper, because we need the protein of the sea, and then the bottom trawling is still not banned. But it's changing everything, also the biogeochemical cycles, and also the viable food. And yes, I start to put here, my feet on the trophic ecology of these organisms, because I'm interested to make you understand that the trophic ecology of these organisms may be one of the keys to understand their functioning and to understand the future seascapes. Of course, we have also this drawing, this scheme, in which you have many indirect effects, in the sense of climate change, in which you have warming, you have ocean acidification, you have deoxygenation, you have also changes in the currents, the current patterns that may be transforming, maybe for sure part of the transforming, variables in the marine animal forest all over the world, from shallow to deeper zones, from the Arctic to the Antarctic, from tropical zones to the coral region. But also, possibly, the change in the viable energy, both from the autotrophic or the heterotrophic part. If they are symbiotic, so mixotrophic, they will have a part that is autotrophic and a part that is heterotrophic, or if they are fully heterotrophic because of the heterotrophy of this organism. But things are not new, in the sense that the first question that we have to put in our minds, if this is pristine or it's not pristine, and it's not pristine at all. This is one slide, 18th century Sicily, okay? And these guys are taking one thing from the bottom, which is red coral. For those of you that don't know red coral, it's Corallium rubrum, which is a precious coral. And I want you to see one thing that I tell always to my students. There are here two details that are important to understand. First, okay, I wouldn't make the question, To the students, I make the questions, but not for you. The first is the size. Look at the size of these corals. They are very big. But more importantly, these guys are going with goggles. They have no fins, and they have no weight. So you deduce that these guys were not just diving, free diving at 40 meters. They were probably diving at 5, 7, 10 meters. And all this is gone. After they are gone, and this is very well reflected in this Jackson paper, the oysters in Chesapeake Bay, for example, that they have a specific rule. Now, the people is trying to restore, because we understood that without these huge banks of oysters that were depleted up to 95% of their mass there, everything went wrong. Everything was just disrupted, and everything was just moving in another way. okay? The filtration of the oysters was essential, and these oyster banks were so huge that they could filter the Chesapeake Bay, in a really powerful manner. So one of the things that we have to keep in mind is that everything is deeply changed, and what we are looking and witnessing now is a completely transformed world. We know, but then we have to understand that the equilibria that in that moment was present has, for sure, some rule. And as Marta yesterday said, and I think that she was really on the point, focusing on the point, all these organisms have a rule that we still don't understand in the complex equation of our world, even of the changes that we are witnessing about the climate, probably. So, we are losing an ally, and, well, what we have to do, conservation, restoration, because there is a debate here, and it's an open debate, of course. People is saying, okay, we have to conserve and making this 30%, but we don't have to touch anything. Other people is saying, no, we have to restore. And I would say we have to restore, of course, that may be good, but always base it on science and with a definite protocol, with a definite pathway, not, as crazy, because we are making many mistakes because we don't understand what we are doing, because we don't understand, indeed, how these organisms work, okay? So I will try to just show you what are the paths that I have in my mind and that we are following to just give you an idea of what could be, the solution, one of the solutions, please, that are in this moment on the stake. So the first thing, of course, is understand the environmental factors. We are all trying to understand these environmental factors, the water column factors, from currents, temperature, pH, of course, the transport of particles, and so on and so on. Then for me, it's very important that we understand the trophic ecology and the ecophysiology of the organism. So understanding these points will be essential to make, something like an upgrade, something like make the next step, which in the next step maybe understand the distribution, health status, and the functioning of the system. But without these previous two steps, it's very difficult to just consider what we are seeing. So what I see many times is that the people is studying the distribution of the organisms, and thus in the discussion part of the paper, they say things that may be good, maybe not good, because they don't have tools. It's not because they are ignorant or they are, because they don't have tools to understand what is really the preponderant environmental factors and the reaction of the organisms in terms of trophic ecology and ecophysiology. And then you have the next step. With these tools, you indeed may do conservation and restoration plans. But this is not all. The last step is the most important step, and I will be very, very, let us say, strong with this point. We have to put this on hands of the society. We can no longer, as scientists, put our head in the sand and say, okay, we did it, that's all. You have all the tools now, just manage it. We have to have a compromise with the society in a real bottom-up, let us say, approach to do this kind of program for them. So all the first 4 steps have to be clearly explained and applied with the local people in different areas. So let's talk a little bit about the carrying capacity of the system and the future of the marine animal forest. What is this carrying capacity? Of course, what we are doing when we are making conservation, but also especially when we are making restoration, is, think that everything is something like static, and it's not static. It's a very dynamic system in which we have to take into account what will happen, and if we are making, for example, restoration, if the trophic ecology, and especially the assemblages of the different organisms, are prepared for this kind of, let us say, carrying capacity in terms of everything, in terms of interactions among the organs, in terms of the water column productivity, primary productivity and secondary productivity, in terms, for example, of the ecophysiology, and so on and so on. Having this in mind, we can start making a plan. In this plan, we shouldn't see only the variables of temperature and pH. We are the most evident, let us say, impacts that we have in our organism. Probably what we have to do is also, and I see many of the talks yesterday and in other congresses and in other papers, of course, people that are starting also to try to understand the energy fluxes in this organism to see what will be the future seascape. So we have to go step by step. And also, with this information, we will be able to truly make a optimized, or as good as we can, because optimizer will be never, restoration plan. Not only considering one species, but considering the whole community. This is one of the problems that we have. So restore means, in some way, restore one habitat or one species. Regenerate means to connect all these habitats, seagrasses, seaweeds, coral reefs, mangroves, and so on, as a synergic path. Yesterday I saw one of the talks in which is considered, for example, mangrove and coral reefs. Many people has been working with that, but still the information that we have, I think that is not enough. So you're going from the organism, the single species, to the community, which is very hard. As Marta said yesterday, is very complex, but we should do it in the sense that we should put all our efforts to understand more than we are seeing in a single organism. And all of you will say exactly the same. Okay, give me more money. Give me more money. Give me more people. Give me more, So this is the point. Of course, we need, absolutely, we need more efforts from part of the administration and of the politicians and from the stakeholders, or whatever they are. Because what I can assure you is that these things now are urgent needs that they have. And what I say to Tammy Goulet right now, is very simple, is that, guys, what do you want? We don't have enough data, but because we don't have the power to just implement all these things and understand the most difficult science in humanity, which is ecology, is the most complex, that goes from molecules to communities. And this is why it's so difficult sometimes to have the whole picture. So these are the steps that I suggest. First, of course, understanding the MAF trophic ecology, benthic pelagic cocoon processes, photobiology and photolimitation, the modern care capability to store energy, as for example, Nuria Viladrix said yesterday, is a branch of knowledge that is very important and is still limited in knowledge, the capability to immobilize carbon with these organisms, also the rule of the marine animal forest invasive species, this is also very important, and also make this kind of upscaling calculations. These are only few things that we have to understand. There are many others that have to put on the stake. So when we have this information about what is happening, then we can understand the constraint of the future marine animal forest. For example, what will be the impact of the primary and secondary productivity, because these are suspension feeding organisms. So if the primary and secondary productivity changes, what will happen? What will happen also, with the photobiology and the rule of this photobiology in mixotrophic organisms? We still have to understand why, for example, there are many cues about scleractinians, for example, and how they are photolimited because of the shape, the morphology, the symbiodinium relationship with the host and the holobiont, but also why possibly the octocorals are more successful. And also making experiments in the energy storage capability of the organism to understand this, let us say, translocation to the future generations. It will be enough, it will be enough in front of mass mortalities. The organism will survive. There will be local areas in which we have the opportunity to see that organisms survive, why they survive in this specific area and not 250 meters away. what is happening, what is this micro-environmental part that are just making successful some populations and others not. And also then, with all this good data, make modeling of the benthic pelagic complete processes and how this will change in the future. But attention, without this data, it will be completely useless to make models, because if you know very well, garbage enters, garbage is going out. So the models are very good if they are fed with very good data. And sometimes you read papers and you see the models and say, okay, now I want to know the source of this. And when you see the source, you see, okay, the 1 point of chlorophyll in the middle of the ocean, this zooplankton was taken in the polar areas, and these organisms are in the Caribbean. I don't think this will be really good to make a model, but then they make the good extrapolation. It's not about criticizing the models. It's about criticizing the enormous quantity of material that we need to do the models. And then we can start making this regeneration process based on science. For example, the influence of the ceston transport to the benthic suspension feeding. So where we have to put these organisms, where will be the best way in terms of population structure, of diversity of organisms, or relationship of these organisms. Calculate also the capacity of the system in terms of energy availability, because sometimes you are doing a restoration plan, don't take into account that possibly they are just really constrained by the energy. For example, in the Mediterranean Sea, if you plant a gorgonian now at 15 meters depth or 20 meters depth, probably you will fail, because the rise of temperatures in these critical mass mortalities, the heat waves, is depleting part of the energy that is available for these organisms. And they cannot withstand, because they don't have enough energy to maintain their ecophysiology in summer and especially also in autumn. Also understanding the role of biodiversity and the structural complexity in the success of the restored marine animal forest, and also start to have, implement some questions, for example, integrated multi-trophic aquaculture with restoration, giving the opportunity to take all the energy that is coming from the fish cages, for example, that is going to the benthic suspension feeding organisms, ascidian, polychaetes, corals, and so on, to just have a nursery that is just fed and is just maybe more successful. We did it in the Remedia project, in the LIFE project, not for that, but now we are linking both things to see if we can be successful, but all based on science. And finally, this fourth path, which is the socioeconomic impact that relates to ecosystem services, study the projected ecosystem services in the future marine animal forest, calculate the role of the marine animal forest, for example, in the blue carbon equation globally, understand how restoration is enhancing biodiversity, biogeochemical cycles, and carbon immobilization, and understand how this restored and natural marine animal forest influences the potential ocean biodiversity credits, which is very polemic. And it's a thing that we are just start seeing that is moving everywhere. And if we are not present in this debate, we will lose again the opportunity to be, from a point of view, be very clear about what we can understand and how this can be used effectively or not. We have to just discuss about it. Yes, we are still here. The problem is that we are still here. So we are still trying to understand what is happening. And the other 3 points depend on the first point. If we don't have this, it will be very difficult to just have a clear idea of what will be the future seascape and what can be done. Because if we don't have the basics about the biogeochemical cycles, the benthic pelagic coupling, distribution of the species, ecophysiology, it will be very difficult to have a very good plan for conservation and restoration and understanding what happens. So let me give you one example, okay? Let me give you an example about what I'm talking about. So you have this kind of 4 steps: understanding the MAF trophic ecology, understanding the MAF future constraints, the science-based regeneration program, and then this kind of socio-economic impact and related ecosystem services of the MAF. These are the 4, this is a flux that I understand that has to be made to just progress or made a progress towards conservation, restoration programs of the marine animal forest in general, but also other forests, of course, and other systems. So, the example is basically the capability to immobilize carbon by the marine animal forest, to just maintain and retain this carbon in the structures. Then, second, in the future, look at the role of the primary and secondary productivity, making experiments, okay, of what is happening, and what will happen about this, let us say, this viable energy for the organism. Look at the influence of the ceston transport, okay, and the distribution of the organism and the population structure and the deposition toward the currents and so on, and the repercussions in the associated ecosystem services, in these cases, the blue carbon, the famous blue carbon that we have. So the first is look at the capability to mobilize carbon by the marine animal forest, and this is the article of Martina Coppari, and she made a calculation of how much carbon is immobilized in some way in the structures of the gorgonians each year, okay? And this is something like 0.014 tons of carbon per hectare per year, 3 gorgonians, 3 Mediterranean gorgonians. If you compare with the other carbon that is just stocked during a certain period, okay, you see that the Amazonian forest is 1 ton, wetlands 0.19, seagrass 6.7, and mangrove 1.5. So you are 2 orders of magnitude less. Here is also considered 3 gorgonians, probably considering the whole coralligene will be a little bit different, probably around the wetlands or something like that. But we must do it. And where this comes from this kind of rationale. The rationale that you have primary productivity, this primary productivity is becoming primary, secondary productivity, and also detritus that are going to the benthic organism. These are part of the food that is taken by the heterotrophic organisms, the filter feeders, and this is basically derived some for the growth. Most is for respiration. You have to consider also reproduction, blah, 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 but some is for growth. Not all the growth is permanent in some way. The immobilized carbon will be this carbon that will stand for 1 year, for example, in the case of an hydrozoan, or many decades, hundreds of years, or millennia, depending on the organism that you are studying. So Savaglia, that I said the other day, may have hundreds of years. So you have immobilized carbon there for a very long period. And then the small part that is really buried and maybe just last for millennia or whatever, that will be a minimum part, of course. The second part is primary and secondary production experiments. Let's make experiments. In this case, for example, primary productivity, just in which you focus on the temperature and the pH changes and see the change in this primary productivity. In this case, were coccolithophorics, for example. But what we saw is that there were real huge changes of this organism, of this microphytoplankton, that on the one side increased the quantity of lipids, which is good, because then, but on the other side were much more fragile and much more easily preyed by copepods, for example. So this may be a limitation for this primary productivity. And we must replicate for other organisms, we must replicate for other change and make experiments with other kind of approaches with secondary productivity too. In general, and this is the general concept, is that primary productivity will change. This is Donay in 2006, already just put on the table, and he also predicted that in certain zones it will decrease the primary productivity, and in other zones will increase. This is already, we are already witnessing this in the Antarctic Peninsula, for example, diatoms and smaller. In the North Sea, Kloer and Smetacek demonstrated that the zooplankton during a long time series is also decreasing in size and in associated biomass. So all these kind of things are changing, and this will change, of course, the availability of food for the organism. Then, the influence of the ceston transport and marine animal forest patch structure in restoration. In this kind of approach, we see that, for example, 3 different gorgonians have a different capability to, in the same elapsed time and intensive cycle, they have different capabilities to just capture prey. It's because of the density? I don't know. Density of Leptogorgia sarmentosa, the first one, no, the second one is almost in that study area was 1.5 colonies per meter square. Eunicella singularis, 11, more or less, 11, 12 gorgonias per meter square, and Paramoricea clavata were 36 per meter square. But we couldn't prove anything, in the sense that we didn't make the experiment to prove that. Possibly it's also the position toward for example, the fluxes. Is the population structure? I don't know, but you have to think that this is already one thing that is ongoing, because we have to understand. And Cattell said in this paper, in this book chapter, she started, of course, long before, but she started also to set up the most important concepts about the microenvironment, in which we have to understand that sometimes it's not a forest, it's the first one. It's not a forest, they are single colonies, all in the benthos. Sometimes maybe a forest, an efficient forest for retaining particles, and other times they are not so efficient because they are too, the density is too high, and then maybe the hydrodynamics is different. So we have to take into account this to make this kind of approaches for restoration. And finally, you have this kind of repercussion in the associated ecosystem services and the blue carbon, and here a paper of Dor, which is also here, started to just understand if we go toward a complexity, if we go towards a mature marine animal forest and also vegetal forest, of course, we are going also toward a completely different perception of the biodiversity and the carbon storage. So we think this maybe, is another possibility that has to be considered. And we have to be considered, also in all the marine animal forests of the world. We have to try to make this kind of approach, not only here, but in this examples that we are, but in many other places that we are considering. So we have to understand what will be the marine animal forest capability to immobilize carbon as one of the ecosystem services that are considered one of the important ones related with climate change, for example. So this is what we are doing in Ocean Citizen. In Ocean Citizen, we broke those silos, and here you have people coming from engineers, architects, people that is also physical oceanographers, people that is just working on marine biology, socioeconomists, educators. Many people join together for a specific target in a specific place. Okay, now we will just talk very few minutes about the project. But this is the magic for me. The magic is gathering the people and work truly together for one target and in one place, mainly in one place, okay, and then replicate the results in several places. What is let us say, the steps that we have to follow? The first is shared foundational knowledge. What does it mean? It means basically that we just have a common protocol to apply, a common protocol in economics, a common protocol in education, a common protocol in transplantation, a common protocol, for example, in the water column follow-up of the environmental factors. Then apply this to one main place in which you apply everything, and then 4 replicates: Arctic, Baltic, Mediterranean, Red Sea, and Atlantic. In the Canary Islands, in Tenerife, is the main focus of all this. That is just structuring the project and focusing the project. And what for? We want to enhance the biodiversity, connectivity, carbon immobilization, and the fisheries around this marine forest, because we are not doing only marine animal forest, but we are also just promoting seaweeds and seagrasses, okay, in the different areas. And always take into account local people, not because it's now fashion, because you need to adapt your plan to the local needs. So you have a common protocol, but that has to be adapted to the local needs to have an output. The final output is, on the one side, involve the people in this kind of actions, in this kind of, of perspective, and give the tools also to know the ocean much better, and give the power to the people, to the local people, to make conservation and restoration measures. And this is why I want to thank everyone, because of this other project, the Cost Action Map World, in which more than 330 persons are involved. You are invited to join it, because we have many things there. We have training school, workshops, shorter scientific missions, congresses, fees, and so on, just to gather information, share information, and be, the ones that want to also have this holistic vision of all the planet about the marine animal forest. It's based on trust, transparency, and shared goals. And this may look like strange and even naive. Well, okay, maybe naive, maybe it is. But you have to think that if we don't make this, if we don't make it in this way, we are doomed. If we don't break the silos, if we stay in our own comfortable bubble, it will be very difficult to solve the problem that is the most complex problem that as humans we have faced ever. That is the huge transformation of our planet. So we can go from this, in this model, you change paradigm, and we can go from this kind of conventional green, system to a regenerating process. We can do that, but I think that we have to deeply change our mind and start talking about other things, not only the things that we are putting on the table as scientists. We have to have more implication, direct implication. We have to help the people to understand what is happening, because they don't understand what is happening, and they are afraid. I don't know if you have been talking with people, but my mission is talking with a lot of people, and the people doesn't understand, they don't understand the things, and the other way is that they are extremely afraid, more than ever. For me, it was a surprise in the last questionnaire, in Spain, that 92% of the people were thinking that really climate change is drastic and is really just taking us to the catastrophe. And I was surprised because there you have people for every single party, left, right, center, people of every single, mentality that we may have. So, terms as the growth, there's, as for example, low consumption of goods, just slow down the economy and the consumerism. So these kind of things, I think, that are very important. Not only for the marine animal forest, but in general for everything. So I give thanks to you for your attention, and I'm open to your questions if you have comments or questions.
Thank you so much for such an interesting lecture. I wanted to ask if you upload any type of the data online which can be accessible for any future analysis.
Yes, the project, is obligatory. You have to upload all the data of temperature, pH, the follow-up of the oxygen, the zooplankton. We are still in the beginning of the project. Now, the 30th of June, we just have the first 18 months of this data is not yet uploaded, but of course it's obligatory. We have to upload everything. This is a mandatory for Horizon projects, Horizon Europe projects, so this will be available for, and also, most importantly, the protocols. Because we are doing a toolbox kit book, in which what we are doing is put all the protocols from the water column to historical ecology, passing through economics and conservation, restoration in a single book that can be just, of course, there will be not all the methods, all the things, but in this moment we are more than 75 persons. We are working together in this single book to have, something like just a starting to look for this kind of protocol. Yes.
Thank you so much.
So, Radek Kowalski from Poland. I want to ask you if in the project you have any attempts to use artificial intelligence. Since you will deal with a massive amount of data, I think we don't have a better tool than artificial intelligence to deal with that, to find the gaps, to find the contraordinary results, to find false results, and so on.
We are using artificial intelligence in two ways for the moment. The first is the reefs, because we are using reefs, okay? And these reefs we use artificial intelligence to make the best structure related to the environmental and biological factors of the area. That's on one side. The other that we are applying now is artificial intelligence for recognition of species and biodiversity, especially, for example, for the brood, especially, for example, for all the approaches that we have with fishes and other mobile organisms. Okay, not only to recognize the species, calibrating, but also with the counting of the fishes, because with this in artificial intelligence, I was really surprised about that. It's normal. You can count the fishes, and then you wouldn't count the same fish again, because it just identifies the fish, and then it's outside of the, also for the ROV, this may be. And also, we are looking for the calibrating for the zooplankton. With the Plocan, we are making this kind of proof in which we are using artificial intelligence to count the samples of the zooplankton, not the phytoplankton for the moment, but for the zooplankton. But we are in progress in the sense that, guys, this has to be improved, and this will be probably step by step, we'll have many other implementations of this artificial intelligence, I guess.
Hello, Sergio. Pedro Frade from Natural History Museum in Vienna. Thank you so much for this great talk and your personal perspectives. I have a quick comment and a question. Quick comment. So we need to restore our ecosystems, but we also need to restore our human cultures. And and in places of the world where you work, like in Brazil, I think they're really much ahead in this perspective. We have to regenerate, we have to restore these regenerative cultures of humanity, right? And having that in mind, because if we don't do that, nothing else will matter. We can have the best science, but we need to heal ourselves and our connection with the world. And then the question to you is, I see at the end of your flow that we have to influence people and work with people, but shouldn't we also start with the people that know and how to live in their ecosystems and how to use them? And what is the role for this traditional knowledge and for this other science that is also there that we don't always reach and don't understand? What is the role for traditional science and for indigenous science? And can you tell us a bit about that? How can we? I'm lost. I don't know how to how we can integrate. We
are already doing it all in Brazil. Let me say that very recently, 2023, we published a paper about ethnocartography. What we did is very simple. We went with an ROV and also with Van Vins and look at the substrates, okay? And then we asked to the local people, to the indigenous people that was fishing in the area, if they can do a cartography of the area. It was perfect. The paper is published, it's Pinheiro et al. 2023, and it's fantastic. For two things. The first is more practical, because then you can rely on them, because we don't have money. Again, it's about money, to make a huge ROV campaigns, ROV cruises, ROV surveys. And the second is, but from the beginning, you are just implementing this kind of approach with indigenous people. But let me say that this goes far beyond. I'm working there with several programs in which we, from the beginning, involve people in the decisions and also showing them what is there and hear what is happening. So one of the programs that I'm following and I'm promoting, I'm leading there, is a project in which we understand what there is the marine forest there, marine animal forest, but also seagrasses that are abundant there, and also rhodolites, and then try to make them understand in the near future what will happen, because they depend on the fisheries. And they are so vulnerable. They are so vulnerable. you can imagine, but you come from Brazil, so you can know that. So we are just thinking and, and thinking about all things, and what we have to do is go there directly to the local people and work with them, hand by hand, showing how they can make respectful mariculture, respectful fisheries, conservation, cartography, restoration, from the beginning, and all the initiatives have to be local. I understood that there is no way for this kind of global things. All these COP things, I went to Dubai, I wouldn't go to another one. I don't want, because it's completely useless. So you have to start thinking about working hand by hand with the people, in our case, from the people of the sea, and the local people that listen, and telling them, for example, what will be the impacts of it's not my field, but of coastal erosion. They are not understanding why they are losing their houses, and they are losing their houses. Or why they don't have the possibility to fish in certain areas because it becomes a desert. They have to understand these things, and the only way is, on the one side, of course, understand the processes. We are all here to understand the processes. But the other way, apply the tools that we have to just be sure that they can survive in the near future. And it will underline survive in the near future.
Thank you for your lecture here. Okay, Sylvain from Japan. And I have a question, because you put a lot of emphasis on blue carbon and carbon sequestration, but I've seen the discourse like in moving to this, reducing the importance of marine ecosystem to carbon sequestration. And don't know if it's a positive or negative stuff, because yes, it's more on the focus now, but it's kind of forget the intrinsic values of this ecosystem. I'm wondering what you think about it, like, because now it's only negative emissions and talks about it, but is it good or is it bad?
As far as I understand your question, well, all this is controversial because, for example, coral reefs are considered carbon sources and not carbon sinks, okay? Because basically of the stoichiometrical, equation that we have for the precipitation of calcium carbonate is okay. But on the other side, I would say that a part of this carbon is immobilized for, a huge number of, centuries or even millennia. So at the end, the blue carbon, this famous blue carbon that we are using, we are perverting a little bit this kind of blue carbon about this possibility to use this blue carbon as a exchange money. I don't like it, but on the other side, I'm convinced that all these long-lived structures, also on land, are capable to immobilize a huge quantity of carbon that is around there. So this controversial part that we have on the plate has to be solved among us, but on the other side has to be, in my vision, has to be used to protect this kind of three-dimensional alive structures. Not only the marine animal forest, but every single habitat that is based on these organisms, of this ecosystem engineered organisms. So at the end is this, oh, well, I found that part of this carbon can go to the macroalgae, and then the overall equation is a carbon sink or the other. No. It's a carbon source because also these organisms are releasing carbon. It's okay. But at the end, what you are trying to do is not carbon. It's carbon, biodiversity, biogeochemical cycles, all the things that are involved, all the complex equation that makes these habitats the habitats that we need to just maintain the biodiversity and especially this functioning. And again, in the first slide, okay, the first slide, in all this stress, one of the things is functioning. We still don't agree what function is in the ecosystems. As Marta said yesterday, we still are there trying to understand what we, how we focus this functioning of the system. So making this kind of synergy, I think, is very important, and on the other side, is reliable because we have data about this. Not all the data and not all the processes, but we have this kind of approach.
Hello. Hi, thank you for the great talk. You mentioned the importance of standardizing protocol. However, how do you propose we go about this? And as better protocols come along, how do we synchronize everyone to update, to improve their protocols?
That's a very good question, too. I mean, what we are doing is gather people in each chapter, for example, or in this case in the work packages, and we are trying to use the protocols that we think are the best ones. Sometimes, for example, some of them may be extremely robust, use it for decades, and already, let's say, absorbed by all the groups all over the world. For example, a quadrat, you know, a single quadrat, 20 per 20, of cover, for example. Others are much more complex, as you said. How are you making this? And I think this will be dynamic and progressive. It says that we are putting this on the table, but that doesn't mean that this is like that. And this is not. It is, you know, we can't move anything. No, this will change. One of them probably will be environmental DNA, because it's very fashion and it's fantastic. But on the other side, all the people that is working in environmental DNA, they sell the idea very well, but we're talking with them directly. They know that they are constrained, and yet there are things that are not, that are far to be perfect, okay? The same with other kind of methodologies. So what is the trick? What is the good thing? You make concurrently many protocols that corroborate the overall functioning of the system. Not only one. You are not only using transects for fishes, or you are not only using ROV, or you are not only using the Broove, okay, this kind of tripods that you put on the bottom and film it. You make everything together. And then you see if this may fit or may not fit. And then you decide which is the best protocol. Also, very important, that can be easily replied. Easily done, because if not, you are doomed. In the sense that, of course, oh, thank you, this is a very nice protocol, but it's so complex and so expensive that I could never make this kind of approach to this kind of protocol.
Thank you, Sergio. Thank you for your inspiring talk.
Thank you.
And now we have to go for the group picture. And then the coffee break. I think we can continue this great discussion with you during the coffee break. Thank you again,
Thank
Sergio.
you.